Hi everyone, my name is Rudi. I'm a PhD student at the lab of Dagmar Wachten, which is located at the Institute of Innate Immunity at the University Clinic of Bonn. And today I'm very excited to give you a quick overview on my paper about tissue-resident macrophages in the context of polycystic kidney disease that has been published in the latest special issue on cilia and flagella of the Journal of Cell Science. So let's dive right in. First of all, we wanted to understand the heterogeneity and the ontogeny of the macrophage population in the tissue. And for this, we employed a so-called double fate mapper mouse model, which was developed by the lab of Elvira Mass, one of our closest collaborators. So in a nutshell, this allows us to differentiate between long-lived tissue-resident macrophages coming from the embryonic yolk sac. They all express YFP. On the other side, we have short-lived monocyte-derived macrophages coming from the bone marrow, which are all TD2 positive. And when these short-lived monocyte-derived macrophages now enter the tissue and undergo a phenotypic switch to become long-lived tissue-resident macrophages, they also start expressing YFP, making them double positive for TD2 and YFP. So when we now analyze the renal macrophage population, it became clear that at all time points from embryonic stages up until 1 year, the tissue-resident macrophages, here shown in the red bar graph, comprise the majority of all macrophages in the tissue at all time points. But when we now take a look at the ontogeny, it becomes really interesting. So at birth and at embryonic stages, virtually 100% of all macrophages are yolk sac-derived tissue-resident macrophages, which is shown by the green fraction. However, at birth and 1 week after birth, we can clearly see the influx of monocyte-derived macrophages, represented by the red fraction, into the tissue. But it's also very interesting that the majority of these monocyte-derived macrophages immediately undergo this phenotypic switch to become long-lived tissue-resident macrophages, which is represented by the yellow fraction. But now, let's come back to the disease context. We also have a mouse model which has a hypomorphic mutation in the PKD1 gene, which means that it presents itself with early, slowly progressive polycystic kidney disease. So in wild-type kidneys, we have the normal tubular structures, while in mutated kidneys, we have these cystic structures which over time accumulate, and this then leads to a loss of the filtering function and results in end-stage renal disease. The gradual worsening of the disease is also shown by the increase of tissue weight and volume over time. And in the graph, we can see that after 24 weeks for the first time, we observe a significant increase in kidney volume. Now looking again into our tissue-resident macrophage population, which we just got to know in the previous slide, it is clear and very surprising that they start expanding from 6 weeks on, so before the onset of cystogenesis and before the significant increase in kidney weight. On a spatial level, this increase of macrophage numbers can be observed especially in the vicinity of cysts. Here you can see the example of a 24-week-old kidney, where you can see in the bottom left corner a big cyst, and around it in magenta you can appreciate a kind of ring of macrophages clustering around it. And finally, we wanted to understand the relationship between the tissue-resident macrophages, which we just analyzed in depth, and on the other side, the renal epithelial cells, which are forming the cysts and are the drivers of the disease. So to analyze their quite complex relationship, we basically left the tissue context and turned to a 3D co-culture model, which is a system with drastically reduced dimensionality, which is very well suited to actually study the relationship between distinct cell populations. Which means that in our case, now we only had macrophages on the one side and renal epithelial cells on the other side in our culture model. So when we were now testing our system, we were very happy to see that under normal conditions we see the formation of normal renal tubuli, while under stimulated conditions or when we were inducing cystogenesis, we saw the formation of cysts. Even more so, when we were looking at the tissue-resident macrophages, here shown in magenta, it became very clear that they are not just randomly sitting somewhere in the tissue, they are actually associated with the tubular and cystic structures. Especially in the case of the cysts, you can see they are kind of like wrapping themselves around them. So when we started to quantify the amount of cystogenesis, or how many cysts are forming in our 3D co-cultures, with macrophages coming from healthy 6-week-old kidneys, here shown in light blue, and comparing them to monocultures of just renal epithelial cells, here shown in yellow, it is clear that there's no increase in cystogenesis. But when we now take macrophages from 6-week-old cystic kidneys, so from animals which have polycystic kidney disease, here shown in dark blue, it is clear that we see an increase in cystogenesis. So a similar picture presents itself at 24 weeks. Again, when we take macrophages from healthy kidneys, we don't see an increase in cystogenesis, but macrophages coming from cystic kidneys, again shown in dark blue, we do see an increase in cyst formation in our 3D co-cultures. So now taking all of this together, it becomes clear that the addition of macrophages to our 3D co-cultures per se does not influence cystogenesis. However, it also underlines the complex relationship between our tissue-vessel macrophages and our renal epithelial cells. And it basically suggests that the renal epithelial cells interact and reprogram our macrophages either via direct cell-cell communication or via the secretion of paracrine factors, and this activates the macrophages. And these activated, reprogrammed macrophages then act back on our renal epithelial cells, which now leads to a worsened cystic phenotype and a worse disease outcome. So, and if this little teaser piqued your interest, feel free to check out our paper in the latest special issue on cilia and flagella of the Journal of Cell Science.